Bismillahirrahmanirrahim. Assalamu alaikum. Dear class, I hope all of you are fine and safe at your home. I have prepared this lecture from various research articles. The full information about the research articles is provided over here. So if you want to study more detail of protein microarrays, you can get the full text from the internet, or even you can obtain the full text from me for more studies. Learning objectives or learning goals for this lecture are: we will see first the introduction of protein microarrays, that if there was the DNA microarray, what was the need to discover the protein microarray or use protein microarray. Then, depending upon the applications of the protein microarray, they are divided into three main types, which are analytical protein microarrays, functional protein microarray, and reverse phase microarray. We will study all these three types of protein microarray, and then we will also do the comparison. First of all, we will see the introduction of microarray technology. Microarray technology refers to the miniaturization of thousands of assays on one small plate. It means that instead of carrying out the individual assays for individual proteins, we can obtain the result of thousands of assays together on a small plate. Like we have actually create a lab on a chip. The second name of protein microarray or microarray technology is lab on a chip. Like all the experiments that you individually perform in the lab, you can perform using the microarray on a small plate. This approach was first developed from an earliest concept of ambient analyte theory, and this concept was then it was introduced by a scientist in 1989, and in the decade that followed, this concept was then successfully transformed into DNA microarray. DNA microarray is a technology which determines the messenger RNA expression level of thousands of genes in parallel. Like we can check the expression level of messenger RNA of thousands of genes in a single reaction all together. We use the real-time PCR to analyze the messenger RNA expression level, but for that purpose we need to carry out the reactions of 1, 2, 3, 4 or 5 genes together. But here, by using the DNA microarray, we can get the expression level of messenger RNAs of thousands of genes in a single reaction. However, there are certain limitations of DNA microarray technology, because messenger RNA profile doesn't always correlate with the protein expression. That we couldn't correlate the protein expression with the messenger RNA profile. So, or to overcome this limitation, protein microarray was developed, and this protein microarray was developed as a high throughput tool to overcome the limitation of DNA microarray and to provide a direct platform for protein function analysis. In this diagram, we will see the overview of microarray for genomics and proteomics, and here the difference between the DNA microarray and protein microarray will also get clear. We know that the physiological state of cell is influenced by the external parameters as well as internal parameters. For instance, external parameters include the intake of drug or change in the environmental conditions, and then diseases which are caused by both internal and external parameters. And internal parameters include the genetic changes or the process of aging. So microarray technologies can be applied to monitor the intracellular gene and protein expression level or expression mechanisms. DNA microarrays are used for genetic analysis as well as for the expression analysis, but the protein microarrays are used for the expression analysis at the protein level, and here the expression analysis at the messenger RNA level occur in case of DNA microarray. Now for DNA microarray, some of the DNA microarray are built from tens of thousands of different oligonucleotide probes per square centimeter, that we use the nucleotide probes which are then fixed on the matrix or immobilized on the plate, as well as we have established the high throughput hybridization system which generate huge set of genomic data within a single experiment. Like we can obtain a huge amount of data from single experiment, and then that data is used for the analysis of single nucleotide polymorphism, mutation, and then for the sequencing purpose, and for expression profiling, which has already changed the pharmaceutical research and their use as diagnostic tool. It has a big impact on the medicine and the biological research. As we know that gene expression studies we mostly carry by using the messenger RNA level, that we can check the messenger RNA level by using the real-time PCR or by using the DNA microarray. But as I have told you earlier that the messenger RNA level and protein expression do not necessarily correlate. Means protein functionality is dependent upon post-translational modifications of the precursor proteins and regulation of cellular pathways, which frequently occur by specific kinds of interactions between proteins and or by the reversible modifications, covalent modifications such as phosphorylation or acetylation. So often the protein function depends upon the post-translational modification. All of the protein doesn't go under the process of PTM. So to obtain the detailed information about a complex biological system, information on the state of many protein is required. And the analysis of the proteome of a cell, which means that the quantification of all the proteins and determination of their post-translational modification and how these are dependent upon the cell state and environmental influences, is not possible without the protein microarray or without a novel experimental approach. For this purpose, high throughput protein analysis method was developed, which allow a fast, direct and quantitative detection. And these kinds of interactions like protein-protein interaction, antigen-antibody interaction, enzyme-substrate interaction, protein-DNA interaction, and ligand-receptor interactions. All kind of these interactions, as well as the quantitative analysis of the protein and how it is dependent upon the cell state, as well as the external parameters or internal parameters, the whole data can be obtained from a single experiment, which is known as the protein microarray, which generate a large amount of data which is then analyzed to obtain all of these information. Now comes the types of protein microarray. Immunoassays are the first form of protein microarray, and immunoassays, as the name suggests, that it involves the use of the antibodies. So it takes advantage of highly specific antigen antibody recognition to build a protein detection system. And then the expansion of this conventional immunoassay into the antibody array applications enabled a parallel and multiple detection system by using a small amount of sample. And this sensitive and reliable performance of antibody microarray is a valuable advantage when we study the complex biological system. Around the same time, another type of microarray was developed via the immobilization of purified protein on the glass slide. So to distinguish between the two types of these microarrays, or to distinguish this from the antibody microarray, they are divided into two main classes, one of which is known as the analytical protein microarray and second one is known as the functional microarray. In the functional microarray, we support all the proteins which are encoded by an organism and therefore they are useful for characterization of protein function such as protein-protein binding, biochemical activity, enzyme substrate relationship and the immune responses. More recently, a third kind of protein microarray has been developed which provide an alternative format to analytical protein microarray, in which the tissue or cell lysates are used to form such an array. So mainly we can say that the protein microarray is divided into three main categories, one of which is the analytical protein microarray, and analytical protein microarrays are mostly represented by antibody arrays and they focus on the protein detection. In this class of microarray, targeted proteins can be detected either by the direct labeling or using a reporter antibody in sandwich assay form. So we can say that analytical microarrays of two types, like we can capture the target protein by direct labeling the target protein. First we capture the antibody on the glass, then we add the labeled target protein, or secondly we can use the sandwich type of analytical protein microarray in which the signal is on secondary antibody which recognize the target protein with the help of a tag, and then we call it as the sandwich microarray, and the first type is analyte labeled microarray. The second type of protein microarray, as we have discussed, is the functional protein microarray. And functional protein microarray, as we have discussed, it has broad applications in studying the protein interactions, including the protein binding and enzyme substrate interactions. As you can see, protein-protein interaction, protein small molecule interaction, protein nucleic acid interaction, or kinases, in which we use the cell proteins to be coated on the glass, and then the partner reagent, either it could be a protein or small molecule or nucleic acid or any enzyme, it is added to find out the interaction between the two. The third kind of protein microarray is the reverse phase protein microarray, and it provides a different array format by immobilizing many different lysate samples on the same chip. Like we can, instead of using the purified protein, we are using the cell lysate. That's why we are saying it as the reverse phase protein microarray. And we coat the glass slide with the cell lysate, and then the partner reagent is identified, which is labeled. When it interacts with the cell lysate, we can identify which molecule is making interaction with it. Now we will discuss all of these three methods in more detail. So let's start. The first kind of protein microarray is analytical microarray or analytical protein microarray. The most representative model of analytical protein microarray is the antibody array. And in the antibody array, there are two kinds, as we have already discussed. One is the analyte labeled and other is the sandwich. So first we will see the analyte labeled assay format, in which proteins are detected after antibody capture using direct protein labeling, and successfully found alterations in the protein expression in cancer cell development by using this technique. So as you can see in this diagram that we have captured the antibody on the glass slide, as it is shown that antibody captured, then we have added the proteins which were labeled in different colors. Like you can see the yellow color, green color, blue color, purple color, red color. These are different labeled proteins which are added to the antibody coated glass slide of microarray experiment. And it is not necessary that only one kind of antibody can be coated. We can coat many different hundred of thousands of kinds of antibodies in the same experiment, and these proteins make specific interaction with their specific particular antibody. Like you can see that the green color protein has interacted with the green antibody, blue color protein has interacted with the blue antibody, and red color protein has make interaction with the red color antibody. Then the washing step will be carried out, all the unbound protein will be washed out, and then we can detect the signal of these antibodies, that which proteins are making interaction with it. And by using this assay, multiple differences in the protein expression system can be studied. Then there are different kinds of ligands which are used for analytical protein assays, which have the high affinity and specificity for the antibodies which are arrayed onto the surface. So these different kinds of ligands, these could be the antibodies or antigen, or these could be the nucleic acid aptamers. You can consider the aptamers are the single-stranded oligonucleotides of known length. These are just like primers that we use in PCR reactions. Then these could be the carbohydrates or could be the small molecules. So we quantify levels of different proteins by binding exact proteins to the microarray, like with which they are making the exact interactions. They will interact with the exact kind of like we have seen that exact kind of antibody which was coated on the microarray. In this diagram, you can see the example of analytical protein microarray, and analytical protein microarray contains antibodies with high affinity and specificity that are spotted on an appropriate surface. So this is a chip which is being used for microarray analysis, and we have coated this chip with the specific antibodies which as you can see they are of different colors, means they are specific for different kinds of proteins. These chips are used for proteome profiling and monitoring proteome expression pattern as well as the clinical diagnostic. In this example, which is shown in this diagram, all of the proteins in two biological samples that we want to compare are labeled with different markers or distinguishable markers. For instance, we want to find out what kind of proteins are expressed in the control and in the sample cell. It could be the healthy person or it could be a patient. We have extracted the protein from both of these cells, as we know that there are different kinds of proteins. So these different kinds of proteins are shown in different colors over here. Then we have done the labeling of these proteins. The control proteins are labeled with green color dye or green fluorophore, and the sample proteins are labeled with the red color dye or red color fluorophore. So we have given different colors to different proteins, and then these protein samples are mixed. These two proteins are mixed, and then this mixture is used to incubate with the array which contain the antibody chips. Spores that show up as green or red have an excess of proteins from one sample over the other. As you can see, with these antibodies, the different color proteins are now bound, and they are giving different colors like red color or the green color. So the spores that now appear by image capturing and analysis, as you can see, if the spore is of green color or it is of a red color, these spores show that excess protein of that labeled protein from one sample over the other, while the spores that appear yellow, they show that approximate equal amount of protein from each sample. As you can see that on this antibody, all the protein bounds are red labeled, which are from the sample, which shows that the sample proteins are more on this spot, and all the proteins bound are from control over here, which shows that all the proteins are from control sample. And if you see the yellow color shows that the concentration of protein from the sample and control is equal. As you can see that there are two proteins bound to two antibodies, one of red color and one of which is of green color. So it is giving the yellow color. Yellow color will show that the proteins are in equal concentration, equal amount. And when there is black color, it means that there is no binding of protein to these antibodies. So no control or sample protein has bound to it. So if you see the green color, this kind of color, it shows that the concentration of green sample, like the concentration of control sample, is more, but it also contains some sample proteins bound to these antibodies. And if you see this orange like color, this shows that the concentration of the sample protein is more over here, but it also contains some of the control protein. So this experimental procedure is analogous to the gene expression profiling that is done with the DNA microarray, and it allows the effect of various physiological stimuli and genetic alteration on the protein levels to be examined simultaneously. So in the analytical protein microarray, different types of ligands, as we have already discussed, these include the antibodies. We can quote it with the antibodies, antigen, DNA or RNA or oligonucleotide aptamer, carbohydrate or small molecules having the high affinity and specificity. They are spotted on a derivatized surface, so to find the interacting partners of these proteins. There are certain limitations of this method, which includes that this method lacks the specificity in protein target labeling, and it has a poor sensitivity for low abundance protein. Like if the proteins are very low in concentration, so those proteins are as compared to those proteins which are high in concentration, they face the problem of specificity and sensitivity because they are not able to interact with their target labeling. So the one limitation is that this method lacks the specificity in protein target labeling. We are labeling all of the proteins with single dye, as we have seen over here. And second is poor sensitivity for low abundant proteins. Like the low abundant proteins, we will be unable to identify the low abundant protein. Second limitation is that the targeted protein labeling may lead to the epitope destruction. Epitope is the site with which it will make interaction with the antibody or the molecule which is captured on the chip. So target protein labeling may lead to the epitope destruction because some of the chemical reactions may alter the surface of the protein or the target proteins. So to overcome the limitation of probe labeling, that less specific labeling, another format of assay is developed, which is the sandwich assay format. And in this format, it provides the higher sensitivity in comparison to the analyte assay format. And in this format, two different antibodies are actually employed to detect the targeted protein. One of the antibody is known as the capture antibody, which is immobilizes the target protein on the solid surface. So this is the capture antibody which is immobilized on the solid surface, and this capture antibody bind with the specific protein, while the other antibody is known as the reporter antibody or detection antibody, and this antibody generates a signal for the detection system. Using the two antibodies specifically and significantly increases the specificity and sensitivity of the sandwich assay format, and it is able to detect the concentration of the protein at the femtomolar level. So both the limitations of the analyte assay format can be overcome by using the sandwich assay format. But In addition to those limitations that have been overcome, there are certain challenges that are faced by the or analytical protein microarray, because the major challenge is the need of high specific antibody. As we know that antibodies are the most popular protein capture reagent, although their affinity or specificity can vary. For example, many proteins or many antibodies can cross-react with the proteins other than their expected target proteins when they were tested on the functional protein microarray, especially when multiple analyte detection is employed, like we have used the multiple antibodies, we have immobilized those or spotted those antibodies on the glass, then there is a chance that they may cross-react with proteins other than their exact target protein. So there is a need for highly specific antibodies, and it becomes a major challenge in the use of analytical protein microarray, because the non-specific binding will lead to the large number of false positive results. Another challenge arises from producing a large number of antibodies in a high throughput fashion. Like we need to produce antibodies in large amount, and if it is possible to produce a large number of antibodies in a single way, like instead of first purifying and making one antibody and then the other, if we carry out the production of antibodies at a time. So the solution to this problem or challenge is the recombinant DNA technology. So recombinant antibodies have become a promising means of overcoming this problem. However, they must overcome the complex fabrication issues, for example, the cloning and expression, before they become the practical for use in today's research lab. Second kind of protein microarray is functional protein microarray. The ability to characterize the protein functionality at the proteome level, it has been highly desirable for modern proteome as well as the system biology studies, because functional protein microarrays are constructed using the purified proteins, and they are constructed by immobilizing large number of proteins to assay the enzymatic activities and also to detect the antibodies and other functions of the protein. So the main purpose of functional protein microarray is to find the functions of the protein, and it enables the study of various biochemical properties of the proteins. Like by using this technique, we can find out with which molecules protein can make interaction, and for this purpose we are using the whole protein or peptide which is spotted on the matrix, and then we do the immunoassay, which involves the antigen antibody interaction, or if we want to check the protein interactions, or may protein ligand interactions, interaction of the protein with the DNA, interaction of the proteins with other small molecules, or interaction of the protein with the peptides, or if we want to study different enzymes like kinases which carry out the phosphorylation of the substrate, and we also profile the immune responses. So the first use of functional microarray was demonstrated in 2001, in which the substrate specificity of protein kinase was determined in yeast. Moreover, the protein microarrays enables the study of many post-translational modifications like phosphorylation, acetylation, ubiquitination, S-nitrosylation in a large-scale fashion, and it is critical for understanding the cellular protein synthesis as well as the function of the protein. So these are mainly the applications of functional protein microarray. Like we can say that the antibody array can be used for clinical diagnosis or environment or food safety analysis, and this antibody is actually the complete protein if we consider it functional protein microarray. Then The functional arrays which are mainly used to study the various type of protein activities that we have discussed, like protein-protein interaction, protein-lipid interaction, protein DNA interaction, protein small molecule, or we can say that the drug interaction, protein peptide interaction, identify the enzyme substrate, and to profile the different immune responses. So the functional protein microarrays, they require the native protein or peptide. So the major difference between analytical and functional protein microarray is that in analytical protein microarray we can use the antigens or antibody or different types of ligands to be spotted on the glass, which can be DNA or RNA, aptamers, carbohydrates, or small molecules with high affinity and specificity. Those are spotted on derivatized surfaces. But the functional protein microarrays require the native proteins or peptides which are individually purified, or they are synthesized from the cDNA, or by carrying out the in vitro transcription or translation or other high throughput techniques. After the purification and their spotting, they are spotted onto a suitable surface, and it will form the functional protein microarray. These chips are then used to probe the protein activities, their binding properties, and the post-translational modification. So the main purpose of functional protein microarray is to find out different activities of the protein, their binding properties, which properties they possess to bind to different molecules, and we can also study the post-translational modifications of the proteins also. So in this example we will see that how the proteins are used in functional protein microarray. So first of all, the protein is extracted from the sample. Then comes the process of labeling of the protein. Proteins are labeled with distinguishable markers. For example, here the proteins are labeled with green fluorescent dyes, and then they are allowed to hybridize on the chip, and the protein chip contain different peptides or protein sequences, a full protein, which are spotted on the membrane. So labeled proteins will hybridize to their specific partner proteins, and after that the spots that light up in the result, after image capturing we will analyze the result, and the spots that will be light up, these are the candidate binding partners. Like the proteins which were spotted at these places, they have found their partner, and it is shown by the green color, and the black color shows that there is no binding partner for these protein. So with the proper detection system, functional protein microarrays can be used to identify the substrate of enzyme. In this way we can identify the substrate of an enzyme, and this class of chip is particularly useful in drug and drug target identification, as well as in unraveling the different biological pathways. Employing the opposite format to classical protein microarray, reverse phase microarray expands the application of this technology. So reverse phase microarray is the third category of protein functional microarray, and this method allows for the analysis of many samples which are obtained at many different states by directly spotting the tissue, cell lysate, or even the fractions of the cell lysate on a glass slide. So in the first step, what we will do, we will spot the tissue or the cell lysate or the fractions of the cell lysate on a glass slide, and then the primary antibody is added, and this antibody is specific to any one of the proteins which are present in the cell lysate. And for instance, as you can see over here, this is specific to this blue color protein, so this primary antibody will interact with this blue color protein. Now we have not labeled this primary antibody, because for its specific detection we will use the secondary antibody. Secondary antibody is the reporter antibody, and it is bound with a certain fluorescence dye, and this is specific for the primary antibody, it will make interaction with the primary antibody, and then this signal will be detected, which is known as the fluorescence based immunodetection. So many different probes can be tested in this way to specifically identify the certain proteins in the cell lysate. But the limitation for this technology is that it can only be used, or it is highly dependent on the availability and specificity of commercially produced antibodies, and because of its this biasness, it has very limited applications. And this technique was first employed in 2001, in which the scientists have successfully detected the different proteins in different stages of cancer. So this technique is used for identification of different kinds of proteins under different diseases. It has only one drawback, which is that the specificity of the commercially produced antibodies. This table shows all the commercially available protein microarrays which are available in all the three different formats, which represents three types of protein microarrays. Some of the companies provide the microarray which is for functional protein microarray or analytical protein microarray, and some of the reverse phase microarray. While some companies provide high content protein microarray comprising hundreds of purified proteins or antibodies, most of them provide a specialized array platform for certain research purposes. For example, for the detection of human cytokines, phosphorylation events, and the study of the certain pathogen host interaction occur. So for the study of the human proteins, prototype array product name, which is produced by the in vitro gene, and it comes microarray. For the kinase study, the Kinex Bioinformatics company provide the Kinex microarray for functional study, and we can identify the 200 human protein kinases from this. We will take an example. For example, the in vitro gene provides a human protein microarray of approximately 9,000 full-length human proteins which are purified individually from insect cell. The quantity and quality of this array is carefully controlled, and the major advantage of commercially protein microarray is that they provide commercial access to researchers who are not familiar with this technology. It means that if you want to identify or you want to perform the microarray, you can commercially carry out the experiment. Like instead of carrying your experiment in your lab, you can provide the samples to that company, and it will carry out the experiment and will provide you the data. However, the disadvantages are obvious as well. For example, the most commercial microarrays do not provide the full coverage of the proteome, like not the full coverage of mostly humans, or only provide the antibodies against a small fraction of the human proteome. And second advantage is that most of them are quite costly. So you need to remember the name of one of the first three, like, or one from each type. Like you can memorize the human type, proto array for functional analysis, and then antibody for specific group of protein. You can remember one group of protein like cytokine antibody microarray, and it is an example of the analytical type of array. Then cell lysate, as we know that it is a reverse phase, so it is provided by the protein biotechnologies, and product name is Somoplex, and it is an example of reverse phase, and it contains a variety of human cancer cell lysate which are used for the identification of proteins. On this slide, we will take an overview of all the types of microRNAs that we have discussed up till now, and we'll summarize the whole procedure of protein microarray. So we can say that protein microarray is actually prepared by immobilizing the ready-made protein samples onto a microscopic slide, which is based on the solid surface using a contact or non-contact microarray that we will discuss later on when we will see the mechanism of how the protein microarray chip is prepared and what method are used for the fabrication. So protein microarrays are generally divided into two major categories, which include the forward protein microarray and the reverse phase protein microarray. Forward phase protein microarray, according to the type of sample immobilized, can be further classified into analytical protein array as well as into the functional protein array. Analytical protein array is a type of array which usually involve well-characterized proteins that act as immobilized probes, for example, antibodies, lectins, and peptides. In principle, analytical protein microarray based assay is equivalent to traditional protein detection methods, for example, ELISA, or Western blotting or immunoprecipitation in a high throughput way. Thus, many different kinds of biochemical assays can be transplanted to protein microarray, which makes them widely used. So the type of the sample for the analytical protein microarray can be the body fluid, cell lysate, and tissue lysate. And it can be used for the diagnosis of the cancer, like cancer early diagnosis and therapeutic assessment, as well as for the discovery of cancer specific biomarker. Actually, here we are seeing the different kinds of microarray with respect to its application in cancer signaling pathway discovery or biomarker discovery or therapeutic predictions. In contrast to the analytical protein array, functional protein microarray usually covers a huge amount of protein on a single slide. Sometimes we can take all the proteomes of an organism, for example, the whole proteins which are expressed in E. coli or Saccharomyces cerevisiae or the humans. So the immobilized proteins on the functional protein array, they are expressed in bacteria or yeast, and most of these proteins are not well characterized. So it is written that some are the uncharacterized proteins which are probed onto a microarray. For this reason, functional protein microarray are suitable for detecting various types of protein activities, and the sample which is used for microarray, it depends upon the identification of those activities. Like if it is a protein, then we want to find the enzyme substrate interaction or the protein-protein interaction. If it is a nucleic acid, then we want to find the protein nucleotide interaction or small molecules, then we are interested in protein glycan, protein lipid, or protein other small molecule interactions. As well as it is also used for screening of biomarkers for certain diseases, like for cell signaling and for the anti-cancer drug targeting protein screening discovery, and for the discovery of the proteins which are involved in DNA repair, and most interestingly identifying the important enzyme substrate, for example, which carry out the phenomena of phosphorylation, acetylation, and ubiquitination. The third kind or the second major kind of protein microarray is the reverse phase protein microarray. It is a special format of protein microarray. It immobilizes the small amount of complex samples, for example, cell lysate or the lysates from the cell lines or the tumor tissues in serial dilutions or the body fluid on a microarray slide. And we use the antibodies for different enzymes or different proteins, and it allows for multiple samples being arrayed on a single slide. Because of this, these samples can be probed simultaneously with the same probe. For example, antibodies detecting the particular epitope, and additional incubation of the secondary antibodies or ligand is required along with the detection and amplification system. In doing so, the target proteins can be traced even using the small amount of samples. So this reverse phase protein assay permits the examination of various members of intercellular pathway. For example, biomarker pattern we can identify in several samples but in a single experiment, and it has been used for studying the different regulation and activation of the signal network as well as the cancer therapeutic prediction. So among all of these protein microarrays available, analytical protein microarray such as the antibody microarray and the lectin microarray have been used most widely in the area of cancer biomarker discovery. Now we will see the example how these three techniques contain different kind of probe on the microarray. We will take one example and what kind of samples are applied on the microarray, which could be labeled, and what kind of information we get from it. So this is the distinctive procedure and representative applications of the protein microarray based upon the discovery of the cancer biomarker using the clinical sample. So here samples that we are using are tumor cells, or serum or other body fluid, and the solid cancer or tissue cell. So first we will see the analytical microarray. For analytical microarray, we need to spot the antibodies on the solid surface, and then the different proteins can be taken, or different small molecules can be taken from the labeled cell. This is the tumor cell, which is the leukemia, and it is labeled, and it can be used as a sample for the analytical microarray, as well as we can obtain different proteins or the different antibodies which can be enriched from the serum or other body fluids, and they can be used as a sample for the microarray of analytical microarray. In case of functional microarray, we spot the different full proteins on the microarray, and these proteins can be taken from the serum or other body fluids. We extract those proteins, or we purify or obtain those proteins by highly expressing those proteins in the host system, and then we coat them on the glass. In case of reverse phase, we are using the cell lysate, which could be serum or the whole body fluid that we have coated it on the glass plate. So this is the sample preparation. We have prepared the sample, and now is the time to detect the signal. When different kinds of molecules make interaction with these samples, different kinds of signals are obtained, from which we obtain different kind of information. So if the serum or other fluid contains certain proteins which make interaction with the functional microarray, we are able to identify the cancer biomarker. Similarly, if these proteins interact with the analytical microarray which contain the specific antibody against those proteins, then we can identify the cancer biomarker. In case of reverse phase protein assay, we are using the different proteins from the cell lysate, and if these proteins interact with the cell lysate and we have identified an antibody, then we can move towards the cancer specific signaling pathway, or we can predict the cancer therapeutics. In this diagram, we will see the comparative and quantitative proteome analysis. In figure A, comparative genome and proteome analysis is shown. Subtractive or comparative strategies which are employed in this method are most efficient for discovering the gene activity in a physiological or pathological context. Equivalent of the proteome, for example, which include the messenger RNA, protein or DNA or phenotypically different sources, are isolated, and then they are subjected to cross-match analysis. These isolated molecules are labeled differently, either with the radioisotope or the fluorescent dye. So here you can see the sample is labeled with the fluorescent dye. These labeled targets represent different phenotypes in two distinct colors. For example, if we are making comparison between the control and sample, then we need to label the samples from two different sources with different fluorescent dyes. One of the sample is labeled with the red color and the other sample is labeled with the green color. Then differences in the sequence or expression pattern of both of these samples can be examined with a comparative analysis using an equal mixture of the differentially labeled targets. For that purpose, the scan of both fluorescent dyes are matched and the resulting color of each spot are characterized. For example, one would expect to see the respective spot colored as the label of treated sample. For down regulation, the label of the control situation is expected. For example, red or green spots which are present in the array picture. Or for the unchanged expression level, an intermediate color of the spot which is shown in the yellow color, it is detected in parallel with the microarray based assay. The same procedure is applied for the parallel quantification of the proteins. Like different proteins are labeled with the fluorochromes and can be detected in parallel with the microarray based assay. Specific capture antibodies are immobilized on the glass slide and they interact with the respective target proteins which are present in the solution. The resulting signal intensity correlate with the amount of the captured protein, like if it has captured more protein or protein which is expressing low. And within each microarray, different kinds of control spots are also included, which include the positive control spot or the negative control spot or the internal calibration spot. It will allow the accurate quantification of the signals which are obtained in the microarray assay. So there are differences between DNA and protein microarray because when we switch from DNA to protein, one has to be aware that DNA and proteins are different classes with different chemical and physical property. Like DNA is built from the 4 different nucleotides which generate a uniform molecule with a well-defined structure and hydrophilic negatively charged sugar backbone. By contrast, proteins are made up of 20 different amino acids resulting in a highly diverse molecules with different abilities. Proteins can be hydrophilic or hydrophobic, acidic or basic, and they are also post-translationally modified. In addition to these, the proteins have diverse and individual molecular structure. Furthermore, the captured proteins must be kept in a functional state when they are immobilized on the microarray. And the antibodies are the most prominent capture molecules, as we have discussed earlier, to be used to identify the target molecules. So there is a need for the development of the different kinds of antibodies, monoclonal antibodies production, which then can be used for identification of the specific proteins which can interact with only those proteins.